Hello, my name is James Lamp, and welcome to my presentation investigating RGS12 as a potential target for cocaine use disorder therapeutics. I'm a freshman neuroscience major at West Virginia University working in research under the Research Apprenticeship Program. With my mentor, Dr. Vincent Totola, and his graduate student, Allison White, we work to find treatments for psychostimulant abuse. Today, we'll be talking about how the knockout of the RGS12 protein can reduce the impacts of cocaine use disorder. Cocaine is widely known for being a very addictive drug. Frequently, users develop cocaine use disorder, or CUD. CUD is characterized by a continued use of the drug despite knowing of the negative biological, cognitive, and psychological effects. It's currently the third most common drug addiction disorder, only following opioid use disorder and alcoholism. It is also common amongst every socioeconomic group and is found often along with other drug addiction disorders. It is also the third most common drug that has been implicated in overdoses in the United States, just behind opioids and alcohol. This translates to cocaine being implicated in over 18,000 overdoses the past 3 years alone. That means that it's been responsible for over 1/4 of total overdoses, according to this graph. This disorder can cause a long and painful withdrawal. The effects of this withdrawal can cause real psychological impacts to the user, including fatigue, depression, anxiety, and nightmares. These effects are typically very severe in their impacts, and they only get stronger if the user continues to use cocaine. Unfortunately, there currently exists no approved FDA treatment that stops cocaine withdrawal. For this reason, drugs with efficacy for cocaine withdrawal or cocaine use disorder are a pressed, unmet medical need. Cocaine's effects are numerous as a psychostimulant. Some of the positive effects that have been associated with cocaine are euphoria, increased energy, and an increased stimulation by already present stimuli. These positive effects contribute to continued usage of cocaine that causes overdoses and cocaine use disorder. Negative side effects, however, are plentiful and can include headaches, weight loss, coma, and even death if an overdose occurs. These effects make cocaine a highly dangerous substance to the user when done in excessive quantities, which is too often the case in modern-day usage. To understand how cocaine exerts its effects, it's important to understand how a synapse works. When an action potential is transmitted down an axon, it ends up in the presynaptic terminal, or the presynapse. This causes the fusion of neurotransmitter-containing vesicles into the cell membrane of the neuron, which makes them release into the synaptic cleft. These neurotransmitters are then free to bind and stimulate the receptors found in the postsynaptic neuron. This is how neurons communicate with one another in the brain. Transporters are an important mechanism of how the brain stops neurotransmitters such as dopamine and serotonin from signaling. These proteins take up neurotransmitters that are found in the synaptic cleft and transport them back to the presynaptic terminal. From there, they are either packaged into vesicles and used for another signal later or degraded. Cocaine exerts its effects by blocking the dopamine transporter and the serotonin transporter. The dopamine transporter has been linked to several drugs, including psychostimulants such as amphetamine and methylphenidate, also known as Ritalin. When cocaine is administered, dopamine transporter is less active because the drug binds to the transporter in a way that does not allow dopamine to be taken up. This means that the dopamine has no way to remove itself from generating a signal and causes the effects that were described earlier. Serotonin transporter also plays a major role in cocaine use. Serotonin transporter manipulation is commonly associated with fluoxetine, also known as Prozac, and MDMA, commonly known as ecstasy. Serotonin transporter blockage by cocaine is a key contributor to the psychostimulant effects that this drug produces. The Yule group, in its 2002 paper, showed that both serotonin transport and dopamine transport blockage are necessary in order for cocaine to show its effects on mice. RGS12 is a gene that is plentiful in the addictive and reward centers in the brain and has been the interest of our lab for the past several years. In the past, our group has run studies showing what effects RGS12 may have upon the dopamine and serotonin transporters and how this might impact psychostimulant usage. Our lab first wanted to examine if the most associated phenotype of cocaine in mice, hyperlocomotion, was expressed in mice that lacked the RGS12 gene. To do this, our group injected wild-type mice and mice lacking RGS12 with cocaine and measured the locomotor response. Wild-type mice treated with cocaine showed a marked increase in locomotion to cocaine. This response was missing in mice that lacked RGS12. Knowing that the dopamine transporter is one important target of cocaine, the lab examined samples of ventral striatum in wild-type and RGS12 knockout mice. This region is rich with RGS12 expression. As seen in the second panel, mice lacking RGS12 much more effectively cleared dopamine than the wild-type mice. To seek an explanation for the increased dopamine uptake activity, our group measured the expression level of the dopamine transporter. Our group found that the lack of RGS12 was associated with a marked increase in the expression of the transporter compared to wild type. Together, these data suggest that a lack of locomotor response to cocaine RGS12 knockout mice is likely due to increased dopamine transporter-mediated uptake, which is a result of increased dopamine transporter activity that is caused by the elimination of RGS12 protein. Soon after, our group ran a similar group of experiments in regards to the serotonin transporter, another key target of cocaine. In a preliminary experiment, we assessed the locomotor effect of MDMA, which specifically targets the serotonin transporter. We saw that MDMA-induced hyperlocomotion was blunted in mice that lacked RGS12. Similar to the dopamine transporter, subsequent studies showed that the serotonin transporter showed increased serotonin uptake activity in mice lacking RGS12, and serotonin transporter expression was slightly elevated in mice lacking RGS12. These models are consistent that with RGS12 knockout causing increased serotonin transport expression and activity. The following is a model as to how the loss of RGS12 causes decreased locomotion in mice treated with psychostimulant. In the normal condition, there are a limited number of transporters that are expressed in each terminal as shown on the left. However, in the absence of RGS12, there is much more dopamine transporter in the serotonin transporter, and the ability to clear dopamine and serotonin is markedly improved. We expect that this increased expression offsets the activities of cocaine and MDMA. Given the data showing that RGS12 knockout mice show blunted locomotor responses to cocaine due to increases in dopamine and serotonin transporter expression and function, we wondered whether these mice would be similarly resistant to the effects of cocaine withdrawal as measured previously by described cocaine withdrawal-associated behaviors. Thus, our hypothesis stated here. Given that RGS12 knockout diminishes cocaine-induced locomotor behavior, we sought to address whether RGS12 knockout diminishes cocaine withdrawal-associated behaviors. For this experiment, we based this procedure on the work of Gasparian, published in 2020. This group concluded that 3 somatic signs found in CD1 mice, digging, grooming, and rearing, were all significantly impacted when a mouse is in the process of withdrawing from cocaine. 21 C57 black 6 mice, 11 of which are wild type and 10 being RGS12 knockout, were randomly assigned to be either in the control group, which was injected with a vehicle, or the experimental group, which was injected with cocaine. The injection of vehicle will ensure that the anxiety and pain caused by injection does not affect the somatic signs of cocaine withdrawal. These mice were injected with their assigned substance over the course of 12 days for 3 times a day. Dosage of cocaine in the experimental group was slowly increased to reflect drug tolerance in cocaine users. After the run of the injection protocol, mice were left to sit for 6 hours in order to induce a cocaine withdrawal. Afterwards, we placed mice for 15 minutes inside an observation chamber to count the number of times that the mice reared, dug, and groomed. Below are 2 examples of the behaviors we monitored for in this study. On your left is a picture of a mouse rearing. It stands up on its back legs for a little bit of time and then it goes back down. It doesn't matter if the mouse is using the cage to help itself rise, just if the front legs are not on the ground. On your right is a picture of a mouse digging. As shown, it is a mouse that is digging into its bedding using its forepaws. These are the 2 sets of data that we generated from the digging and rubbing behaviors of this experiment. As seen here, I have highlighted the wild-type mice's rearing behavior in response to their injection with the red box. There is no significant difference between the cocaine-treated and the vehicle-treated mice. A significant difference would indicate that something has changed in this sign in response to cocaine, i.e. cocaine withdrawal. This result was not expected in our study because Gasparian found that cocaine withdrawal did affect rearing behavior. However, our data does not suggest this to be the case. As such, we are unable to test our hypothesis to whether or not lack of RGS12 affects cocaine withdrawal-associated rearing because we did not observe this phenomenon. Similarly, the Gasparian et al. Study suggested that the digging behavior was affected by cocaine withdrawal. We did not make any observation of this in our data. As seen in the graph, the wild-type treated mice did not show any significant difference with cocaine treatment. As a result, we are unable to use digging to test our hypothesis. The primary reason as to why rearing and digging do not hold any significant difference could have to do with mouse strains. Gasparian et al. Studies CD1 mice. However, our RGS12 knockout mice were of the C57 black 6 strain. Thus, our results suggest that mouse genetic background can contribute to the development of cocaine withdrawal-associated behaviors. Unlike digging and rearing, which were non-informative to cocaine withdrawal, we did observe a marked effect of cocaine withdrawal status on grooming behavior. As you can see here in the red box, there was a 3-fold increase in grooming behavior in cocaine withdrawing wild-type mice. This increase is significant. This means that there is only a 5% chance that this effect was caused by random sampling error. So now we have a behavior that allows us to test our hypothesis as to whether RGS12 genotype affects at least one cocaine withdrawal associated behavior. As can be seen here, there is no statistically significant difference in grooming behavior in vehicle-treated and cocaine-treated mice. Thus, cocaine withdrawal-associated grooming is absent in RGS12 knockout mice. From all of these observations, we can conclude the following. 1. Digging and rearing do not appear to be associated with cocaine withdrawal in C57 black 6 mice. 2. Grooming does appear to be associated with cocaine withdrawal in C57 black 6 mice. And 3. RGS12 knockout appears to alleviate cocaine withdrawal associated grooming. These data show that the lack of RGS12 in our mice has a significant impact upon cocaine withdrawal as measured by grooming behavior. This means that RGS12 knockout, possibly in the form of a drug, can have an impact on other parts of cocaine withdrawal in mice and possibly even in humans. In the future, our plan is twofold. On one hand, we plan to launch a campaign to identify drugs that inhibit RGS12 function. Importantly, no such drugs currently exist. Hence, our use of gene knockout to probe the effect of RGS12 upon cocaine withdrawal. On the other hand, there are likely other somatic signs for cocaine withdrawal yet to be studied by us that may or may not be affected by RGS12 knockout or inhibition of RGS12 by a drug. We're planning to test for such behaviors in future experiments. Our hope is that this work will eventually inform novel strategies to treat the effects of cocaine withdrawal in humans. I'd like to thank the NIDA and the NIH for funding this project. I'd also like to thank the Research Apprenticeship Program and the Department of Undergraduate Research for providing me the opportunity to allow me to work as an undergraduate researcher. Last, but certainly not least, I would like to thank Dr. Vincent Satola and Allison White for their help in developing me as an undergraduate researcher. Thank you so much for watching, and if you have any questions, feel free to leave them in the comment sections below. Thank you.